So tonight I'm going to tell you some exciting research we did last few years, and finally it's published a few days ago, less than 10 days ago, February 10th, published in Communications Biology. So it was the same title. I'm going to give you the QR code near the end. So the question you would ask is, Zach, why are you studying honeybee swimming, right? So because bees do not normally swim. So this all the idea came from a paper published 5 years ago in November on a prestigious journal, PNAS. So in that paper, they discovered bees could swim, flap their wings on top of water. I can show you a video I made. So they have this highly sophisticated camera to show you the wave patterns the bees generate. It's a beautiful photo, but it turns out you could actually see the waveform using your naked eyes. So like this one, I just used my cell phone, and we could see the same waveform as slow motion. You can see the beautiful waveforms. So this is a slow motion, and bees are moving about 3 body lengths per second. It uses very small force by the waves behind it, basically. So right away, I asked the question, how many bees actually fall into water? I mean, for a 50,000 worker bee colony in June or July, that would be when they're most populous. The chances this a bee dropping into water is pretty small. It happens to some water foraging bees, but other bees when they're foraging over a lake, but that's also quite rare. So the chances of this being adaptive is quite small from a colonist point of view. So I thought I need to study it to see if this behavior is actually adaptive, because that study was all biomechanical, how many times the wings flap, how fast does the bee move, but is the swimming behavior directional? Do they move to the same direction, or do they prefer some color when they're swimming? So it's might help their survival. So right away, within a week, I came up with a design like this simple one with a glass bowl, and we could drop a bee in the middle and then see where the bee would swim to. So we give them some sort of a landmark. So this is a piece of laser printed black paper here, which is about 20% of the circumference. So 72 degrees. So the idea is, my original hypothesis was that the bees, if the swimming behavior is adaptive, we should expect that the bees would prefer a dark area. The reason being a dark area is I try to imagine myself as a bee in the water, and I'll try to find a place to land. So a place to land would be a dark piece of soil, a patch of soil, a tree trunk, something would help me get away from the water surface. So that's why black area would be for the bees to swim toward. If bees show a preference toward that dark area, then I would say this behavior is perhaps adaptive. So one way is to put 100 bees here and then show a lot of bees swim toward this black area if you fix the black area in the top. But right away, I questioned my own design. I said, oh, what if people say my design is biased? What if there's an innate preference for the bees to swim to the north direction, or if the room is not, the lighting is not even, so bees are trying to swim to that direction, or the bees could be just trying to avoid me if I'm standing over here in this direction. if I smell bad, they're trying to swim away from me. So they're always swimming to the black. So I have to remove those artifacts. So one way is to rotate the black area uh, 4 different directions. So you move this black area on top to the right side, to the bottom, and to the left side. That way you remove the biases. If you drop the bees, 20 bees here, 20 bees here, 20 bees here, and 20 bees here. That way you balance the 4 directions out. So if you still see the bees prefer to go to the dark area, then you could say it's not a lighting, it's not an innate preference toward north, and it's not my own smell. So it's probably most likely bees are trying to home toward a black area. So I'm going to show you a video of bees swimming. Bees swim quite fast. And as soon as it touched the edge, we would capture the screen and measure the angle. So there's another bee. There's another bee that arrived at a different location. So for this bee, it touched this area. So the next question I face is how do you present the data? I don't want to present 4 different charts showing that the black area is on top, on the right side, on the lower side, on the left side, where the bees landed, because it would be too complicated. So one way is to standardize the black area. So to standardize that, I measure the angle of the bees landing. So I standardize measuring the left edge of the black area, and then the bee. So this angle here, let's say it's 60 degrees. So if I see the angle being 60, I know it's in the black area because that black area is 72 degrees. So it doesn't matter where this black area is. But the problem arises if this bee arrived here, and if you measure this angle, and you say this angle is 55 degrees, and you would misrepresent the bee still in the black area. So therefore, you should measure the large angle. So you should measure the left edge, then clockwise, you measure this big angle is actually 290. So 290 is larger than 72 degrees. So you will know the bee is outside that angle. And with this data, you could actually present a scatter diagram like this. The bees are landing in different places, and each small dot is a bee. So here you have 5 bees landed here. You have 6 bees landed here. So, and you could see right away there's concentration of bees landing in the black area. So we had all 4 directions, the black area put here, located here. And then you see more bees are concentrated, and there's a statistical test called the relay test that tells you if the distribution is random or not. And so this is control bees that are fed with sugar syrup with a small little bit of acetone to control the solvents that were innately used for giving bees poisons. So we have 3 replicas, they're all the same. So very close, mean angle, the so the mean angle is smaller than 72 degrees because the bees are concentrated here. But another way of testing this is to see, because the problem with relay test is people could ask, well, what if the bees are avoiding the black area? You would also show a non-random distribution. Of course, you could argue that by eyeballing, bees are more concentrated in the black area. but you have to prove it, right? So one way to prove that is to use a chi-square test. Is you divide the circumference by 5 blocks because the black area is 20%. So you have 5 sections. Each one is 1/5. And then you know how many bees landed in the black area. Yes, 40 bees, but the expected number of bees is actually 17 because 85 divided by 5 is 17. So then you calculate the percentage and you do what's called a chi-square test to see if this pattern is random or not, if the distribution is all equal or not. If it's all equal, this p-value should be higher than 5%. So this is about 0.2%. So that means it's highly likely not due to random. So bees are actually preferring, it's almost 50%, almost half the bees landed in the black area, even though the black area is only 20%. So this is not a test. So it happened at that time I had poison bees in study that I could use right away. So the reason I want to use intoxicated bees to test is if I'm lucky, the intoxicated bees would show a random distribution. That would further strengthen the argument that bees, the original preference to dark shown by the normal bees must be adaptive because the bees now when they're intoxicated, when they're drunk, they could not show a preferred direction, swim to the black area. So it's a supportive argument for showing that it's a cognitive process. So it happened that paper was already published to compare the same level of pesticides, but bees stationed at 2 different temperatures. So one is at 125 ppb thermotoxin, but at 24 degrees, they all died. That's most bees died for imidacloprid. But for thermotoxin, also more than 50% bees are dead. But at 35 degrees, when bees are normally clustered, when they have brood, the mortality is quite low, less than 20%. So we had those bees left over, we could do the swim study, so that's what we did. Those bees are already dead after 5 days. So we use those bees, and right away you see there's no concentrated bees in the black area. We have one trial done in Michigan, one done in China, and this one has many more bees. So you see one audience asked me the other day, well, Zach, you see another bees here outside. But I say you need to compare the bees in the dark area compared to outside. So here you see bees here outside, but also inside the black area. But overall, it's actually random. So the mean angle is actually close to 180 degrees. This one's almost exactly 180. So when it's 0 to 360 degrees around, the average should be about 180 if it's totally random. So this one's close to that. And indeed, René's test is not significant. So it's totally random. And if you do a chi-square also would show the same thing, that acetone bees show a preference to dark, whereas thermotoxin toxic poison bees would show the same as expected, about 20% of bees landing in the dark area. It's almost the same as calculated expectation. So then I was puzzled for a while. I said, how do we know if the bees that are poisoned, if they are affected, their cognitive process could be affected in 2 different levels. One is they get so intoxicated they did not know they have to go to the black area. they forgot. Another one is they still knew they want to swim toward dark area, but the muscular control, they lost the muscle control, so they couldn't go to the black area. So a common sense scenario I imagine is when the cops stop a car driver suspecting the driver is drunk, and they do a test. They ask you to walk a straight line. But if a driver is so drunk, he doesn't know what walking a straight line is. He will refuse to do it. So that's totally drunk. Another one is tried to walk a straight line but couldn't balance. So I'm failing to walk a straight line. So those are actually two different cognitive levels. One is actually more intoxicated than the other. So, and it turned out we actually have data. So we have the video of bees, how they reach the edge. So we track the trajectory. We could figure out how many terms are made. So for example, this bee made 18 terms in the control. But by and large, a lot of bees had 0 or 1 terms. So the average is 3 here for the control bees. For the pesticide poison bees, I made 9 terms, 9 circles when they're trying to swim. So this suggests me that the bees lost some muscle control because I made 3 times more terms. It's possible they could still forget the need to swim to the black. But at least we know they have lost their muscle control, which makes sense because neonicotinoid is a neurotoxin that affects basically their muscle control and also their central nervous system because they bind to the acetylcholine receptor and causing spasm. So then the other question I asked is, well, because we imagine that it's hard to argue that the bees have this behavior because social bees, it's very small cost to lose some bees that are drawn in the water. So I thought maybe most insects might have this preference to dock already pre-wired. So how do you test this? One way is to test some solitary bees that are still a bee but do not have social behavior. If they show the same preference behavior, or even better, if they have stronger preference to black area, then my argument would be correct. So lo and behold, we have mason bees, males and females. They all show very strong preference, almost I think 80%. So 70% of bees go into the dark area compared to honeybees 50%, about 70% here for both male and female. My original hypothesis is maybe the males are more dumb and be not as good as the female bees. But they actually show the same preference. But I was partially right, but the female bees actually did swim better. They had faster speed, shorter duration in their swimming behavior, even though the preference was also dark. So for example, the duration here, the female mason bees is shortest, shorter than honeybees. Honeybees shorter than mason bee males. And the distance, again, mason bee females and the males and honeybees are the same, but females are shorter distance compared to the two groups. Actually, males are also shorter than honeybees. And velocity, so the speed, the swimming speed is the fastest, so the higher is faster, 2 centimeters per second. So female bees faster than honeybees, honeybees faster than male bees, male mason bees. Actually, no, honeybees and the female mason bees are the same. This is NS, non-significant difference. So those two are the same. But honeybees are different from mason bees and different from female mason bees and male mason bees. Very confusing. So basically, the take-home messages is that honeybee swimming behavior or bee swimming behavior, because we have both honeybees and mason bees, are not random because they home toward a dark area. And then the pesticide changed this adaptive behavior, so landing became random. So basically, we have another harmful effect shown by insecticide that because if you have bees foraging in a bucket of water and for some reason they get blown into the water surface, they are less likely to make it back if they have been poisoned by pesticide. So I had a high school student also did compared 4 species of bees in China last summer. My hypothesis was that open nesting bees, which is giant honeybees, Apis dorsata, and dwarf honeybees, Apis florea, those two groups of bees should have perhaps a stronger dark preference compared to cavity nesting bees. So we happen to have Apis mellifera and Apis cerana in China. I always want to have a replicate when I argue something like this. So you have 2 species in each group. And then it turned out I was right that the cavity nesting bees, those two here that domesticated, show a weaker dark preference compared to the more basal honeybees. So those bees are more closely, more similar to mason bees, their swimming behavior. Oops. So I have the QR code here for the paper, and my websites are here, beephotography.com, and my honeybee research website at Michigan State is bees.msu.edu. With that, maybe we'll leave the question at the end after Megan finishes. Thank you.
So I'm glad Zachary presented some data. I don't have a lot of data to present. I just want to give an update of some of the projects that we're working on. One of the things that we talk about is if we don't share all the different projects we're working on, it's hard for everyone to know just how many things we're doing. And I feel like we have a billion things going on, and it's nice that everyone is aware of all of those things as well. So the bulk of my research, a lot of it is still focused on European fall brood, looking at how we can prevent it, how we can treat it, how we can control it. So I have a lot of upcoming field work on that. And then I've been working really closely with Peter Fowler, who is now in my lab as a postdoc. And we've got quite a few questions regarding European fall brood. So we just recently published a paper looking at prevalence. We're interested in the transmission and prognosis, how it's treated. And then we've started to do some work on what kills it. And then we're going to continue doing more disinfection studies this summer. And the reason I'm not presenting a lot of the data is that he and I recorded about an hour and a half video that's on our YouTube channel. So, Michigan State University Beekeeping YouTube, and we host European fall brood office hours. So, we've done one the last 3 years, and they all build on each other. It's basically the update of what we've done with European fall brood. The bulk of what he and I have been able to kind of figure out the last few years that we've been working together on this is really, really high prevalence and very high co-infection. So that means co-infection between the two groups of strains. So we have like atypical and typical European fall brood. We did a prevalence study in Michigan, and we found that it's in about 20 to 30% of colonies. It's very highly seasonal. So we see a lot more of both the disease and the levels of infection in June, but we're able to basically find it all season long. But I was really surprised at how just the level of disease. So we have much more disease than I think anybody anticipated. Our levels are kind of in line with the national levels, but it's really, really high overall. And then in certain situations, like if we just focus on these high risk scenarios, like in commercial hives after blueberry pollination, right in the peak season, we were finding levels as high as 60%. And in some operations, we'd find like 90%. So really, really high levels of European fall brood. We have been looking about whether or not the antibiotics still work. So OTC represents oxytetracycline. And we published a paper demonstrating that the strains of European fall brood that we have circulating in the United States don't show any signs of resistance to oxytetracycline. There were two papers, one from Japan and one from Canada, that kind of indicated that it could be likely, or it seems to be that maybe it was. But what we showed and what this figure's kind of referencing is that a lot of that has to do with the methods and the different type of media that those strains were grown on. So the bacteria that causes European fall brood, Melissococcus plutoneus, is very, very, very difficult to grow. And we have some methods to grow it that were developed decades ago, but they're not great. So when we grow it, it's kind of grown under duress. And the different conditions really affect the rates of growth, which really affect these different assays. So it's hard for us to say, oh, this is resistant or oh, this is growing well because it's still, we haven't perfected how to grow it. But the reason that we think that it's not resistance or that it's unlikely to see resistance is because we also were able to show that it remains viable on surfaces for years. And the way that it does this is probably forming a biofilm. So what that means is that when the bacteria are drying out, they exude these little strands of DNA that kind of form this protective shield over it that allow it to remain on the equipment for years. So a lot of people have known that American fall brood stays on equipment for years because it forms a spore. European fall brood, we were able to grow it out of a dried larva off of a slide like 3.5 years later. We can grow it off of different like wood and steel, like representing different hive components at least 2 years later. So it stays on equipment for a really long time, probably because of a biofilm. So we're finishing up some studies with this as well. And so obviously this is really, really, really important to beekeepers because that means that there's a much higher risk as you're moving equipment around. But this is also probably why the antibiotics still work, is most of the time when the antibiotics are in contact with the bacteria aren't replicating, which means that they're not really affected by the antibiotics. And the antibiotics, oxytetracycline, breaks down really quickly. And so the bacteria can just wait it out. So it's under pressure. It still works in cases of active infection, but then once the antibiotics wear off, the bacteria is just kind of hanging out there. So it doesn't get rid of it from the operation. And I can't see the Q&A right now, so I'll do questions at the end if anything comes up. So we aim to keep working on European fall brood. I mentioned that Peter is staying on as a postdoc. And we did, so when we did our prevalence study, we actually sequenced a bunch of strains using, it's called whole genome sequencing. So we have the whole genome of, I think, 92 different isolates. And we found some really interesting things in those. So we are working on a manuscript describing, and a lot of it has to do with the viruses that affect the bacteria and then the bacteria's immune system fighting off those viruses. So there's some whole dramatic elements as part of the genome that we're looking at. Notice that I'm saying partnering with bioinformaticians because I'm using we really broadly. We put in a grant to try to put in for a phage therapy center. So that would be trying to find viruses that would kill the bacteria, which would be awesome. This is Jess in the photo here, and she is a PhD student who is in the molecular biology, immunology, and genetics department. And she is doing larval rearing, looking at the virulence of all the different strains. And so we did this thing where we went out and found all of these different strains in all of these different hives. We found a lot more diversity that we anticipated. So now we're trying to see what those differences mean in terms of virulence and how they affect the larva. So she is gearing up to do a lot of larval assays where she raises the larva in these little dishes that you see here in an incubator, and then will grow the larva up, feed them different strains, and look at their different outcomes. And then we're continuing our disinfection study. So our data from last year show that bleach works really well, which makes sense because that would affect the biofilms, and bleach tends to kill a lot of bacteria. Alcohol doesn't work as well. And we looked at it in the context of wood and steel, so like woodenware and hive tools. We haven't gotten to the point where we have a recipe and we'll say use this amount of bleach in this amount of time. We were doing 10% bleach for 10 minutes, but we haven't tried it on like full scale equipment. So that's what we're hoping to do this summer is looking at the virulence of the different strains, and then to continue looking at different disinfections. This is the project I'm working on literally today. So this is Katie Lee from the University of Minnesota, and she and I got funding through Project APISM for last year, and then we actually got funding with Dr. Rogan Tokach as well through the North Dakota Department of Ag to continue this for another year. But we're working with a commercial beekeeper out in North Dakota, and we did 100 and no 240 hives that we've been following since May. And this is their graduation weekend. We're going to see how many of them made it to almonds in good condition. And we did kind of three different treatment trials. So we did from May to June. We had a treatment in as the colonies were building up. We had treatments in through June and July, and then we had treatments in after they took the honey off. And then we're going to see how they went. So from May to June, we compared amitraz to formic acid. We don't have the results yet because, like I mentioned, the studies aren't completely done, but definitely the formic acid did quite a bit better in the beginning. So the colonies that received formic acid compared to amitraz in May did better overall, regardless of what the rest of the treatments they got through the whole year. The other thing that we saw is that doing three treatments, so one in May, one in June, even though it was a long period one, and then one in August, was probably not enough in this condition too. So those summer treatments are really, really, really important. But we hope to have data, from this year, and then also we just got funding to start another year of data. And as part of that, we're expanding to work with Michigan beekeepers too. So we'll keep the North Dakota, California route and then do a southern US up to Michigan route as well. We have been participating a lot with the USDA lab in Baton Rouge, looking at these special boxes that have extra levels of propolis on them. And then also looking at pole line bees, so trying to find some things that can make bees more resistant to infection. So that was the first year was funded through the Eastern Apicultural Society grant, and then we did a second year based on other little bits of funding that we can put together. And that this is still all being written up by Joe, who you can see here from the ARS lab. Another project that we're looking at is kind of along the line of the disinfection, but also as a way of varroa control and ultimately potentially as a way for tropicolaps management, is to look at shook swarms in commercial operations. So we had a student a few years ago work on a project using shook swarms where you take the bees and you shake them onto brand new equipment as a way to manage bacterial disease. It's pretty commonly used in Europe to manage foul brood diseases, and it's used occasionally in like small scale operations, but it's not often used in large scale operations. And so we're evaluating the effectiveness of doing this in large scale operations, and then also just showcasing people who are already doing this at scale. It's a nice way to manage disease and to create a break in the brood cycle. Some fun non-disease related grants. We have one that's finishing up this spring where It's working on bringing more native seeds to Michigan. So we actually currently have a national native seed shortage, and we also have a Michigan native seed shortage. So it's awesome that people are wanting to put in more land into native habitat, but we don't have enough seeds in Michigan that are from Michigan plants, or people really want them to be Michigan genotype. So one of the projects, this is a SARE partnership grant. We worked with the native plant growers and we helped them get access to some of the rarer plants or ones that are a little more difficult to grow or new for them to grow. And then we also are training farmers to grow native seeds that they can sell on consignment to these other native plant producers. So the idea is that we've got lots of growers in Michigan. A lot of them have hand labor. A lot of them have extra space. So here you can see a blueberry grower who has extra space and they're going to grow native plants in between the rows and then sell the seeds as like an alternative source of income. And then we're also working with a market vegetable grower and then also with some plantings on MSU's campus as well. I know some of you have heard about this project. This is Maple, our honeybee disease detection dog. So her handler, Sue Stayskull, and some of us at MSU are working on creating a guidance document to train dogs to identify diseases in honeybees. So a big part of it is actually trying to get the dog disease or the bee suits to work for dogs, which is a very cute and fun project. And then really how to, the biosecurity conditions, like how to do the actual trainings, how to safely get samples, things like that. So that project is continuing through the winter, and we hope in the next couple of months to actually have a published training manual out for people. And then I'm just working with other parts of the university. So we've got a couple projects going on. This is Francini, who is in the Department of Animal Science and is our poultry specialist. And she and I got a project looking at how to use propolis as an alternative to antibiotics in turkey production. So it's actually a feed additive. And then this is Mason McNair, who's from the Department of Horticulture. And he's really into plastics and 3D printing, and we've been experimenting with different plastics that don't require a wax coating for foundation. And so we're starting year 2 of both of these projects this coming summer. And then finally, this is the last year on our grants where we're doing the training for veterinarians. So I'll continue to teach at the vet school for veterinary students in honeybee medicine. And we'll probably still do a couple workshops, but we had a really large funded multi-state, multi-year training effort to get veterinarians trained in order to take honeybee medicine. And I know at least we definitely have veterinarians in the state of Michigan now around the state that will take on honeybee clients. So we have a list of them on our website, which is Bees Need Vets. And I'm happy if someone's looking for a veterinarian to work with, I can definitely pass along a list for you. And so we'll probably do some stuff, but those grants are winding on down as well. All right, so here's my contact issue if people have questions, but then I can also stop sharing and we can pick up some in the Q&A.
Thanks, Megan. So, Zach, I'll start with you. There's a comment or question that says, "Thank you so much, Dr. Huang. Just to clarify, was it able to be determined if the pesticides affected the bees' pathfinding ability to navigate to the dark spot, or is it instead affected their muscular ability to get to it?"
We can't say the other parts does not exist, but we know at least the muscle activity is affected. But we actually, the other questions we do not know the answer to. Yeah. It could be affected. For example, the bees could forget the need to swim to a dark area, or they still know the need to, but the muscle control is lost, so they can't get into that. And those two we cannot actually distinguish. By the way, do you know the muscle activity is affected? And make them make more terms. Yeah.
Thank you. Megan, will using a bleach solution minimize European foulbrood?
So yes, probably, but we don't know exactly um how, and it sounds like it's such an easy question. And I can tell you when I wrote the grant, it felt like a very easy question, but anything that makes sense and is straightforward, the reason it hasn't been done is because it's way harder than you would think it would be. So the issue that we have with this bacteria is A, it's really hard to grow, and the different strains grow at different speeds, but also it's really hard to quantify because it clumps together and then it shoots out excess DNA. And the way that we normally quantify bacteria is by doing PCR, by like counting the amount of DNA. But with this bacteria, you can just count a bunch of DNA, but it's not actually alive. And so when we're looking at a disinfectant, we want to know how many alive, like what is the threat? And so we spent about a year just trying to quantify bacteria. So it's super fun. If that appeals to you, come join my lab. But it's really hard to tell viable bacteria. So we've gotten to the point where like we will take the sticks and we'll dip them into bacteria, the like in a broth solution, allow it to dry, put it in the bleach, and then we can rinse it again and see how much is left on it. The other thing that's hard with ESB that, collectively nobody can do is set up a challenge experiment. So what a challenge experiment is you will take like a hive and add the bacteria and then see if they get sick. And part of it is because it clumps up and because it dies so quickly, it's hard to like spray it on. Like we can't take a hive, spray it with bacteria, and then it gets sick. And so we can't, that's the study you would want to do to see if you could use the bleach in the field. So we're going to keep working with bleach and come up with some recommendations. We've also got some fumigants. We're going to look at ozone. We did already look at UV and hydrogen peroxide, and those didn't work well. And then, so we're going to also look at some classic veterinary disinfectants as well.
Great, thank you. And then Megan, could you please explain the shook swarm practice?
Yes, I did kind of go over that pretty quickly. So with a classic shook swarm, the idea is to take the bees and to put them on brand new equipment, like foundation. And we actually do this, and I personally have done this effectively for American fall brood, and this is considered a completely acceptable way to manage a colony with American fall brood, as long as there's enough time in the season for them to rebuild. So let's say you came out to a yard, you had or and this could be with a single sick colony, but what we're looking at it as a method that you would just kind of do this regularly as a way to minimize disease. So in either case, you would take the original colony and you would take the bees and shake all the bees onto brand new equipment. If it was American fall brood, you would take those frames and burn those frames and destroy them. But if you're doing this as part of a practice, the idea is that you would, shake the bees onto new equipment, and then you'd have a bunch of brood frames, and, you could have some of the bees on there, and those could be other hives, but you would be able to separate your operation into hives with brand new equipment and then hives with the old equipment. So in the commercial scale, one of the beekeepers that we're working with, what he does is shakes packages off of his own bees, puts them in these big holding cages basically, and feeds them a lot. So then they're in a cage with no combs, so they can't regurgitate anything, which forces them to swallow and eat whatever's in their guts, and then it'll make a lot of wax. Then the next day, they'll put them right onto wax foundation, and again, keep feeding them and install a caged queen. So you've got a really solid break in the brood cycle. You've got a brand new queen. Everybody's uniform. And this one beekeeper does, I think, 400 and some hives at a time. So that you could, you can do this at scale. And then the colonies that are older are the ones with the old equipment. We call them the mother colonies. So they have all the old brood and they still have the old queen. Those go off to pollination and they get harassed and then eventually they probably will get sold. And then all these new colonies that have fresh wax and young bees and a young queen and a nice big break in the brood cycle, those bees go off to honey production. And so it's a way to kind of minimize the disease and keep track of where your old equipment is. So that's the theory behind it. Okay, and I do see the question about the hydrogen peroxide. I'm not sure exactly which strength solution. We did publish that paper, so I can use that. But I think part of that is because of the biofilm formation. It was um, less effective than the bleach would be. You have to have something that will really break down the structure of the biofilm.
Great. Well, those are all of our questions. So thank you everyone for joining, and thank you, Megan and Zach, for sharing with us.